You guys hear me now? All right. You still okay? Can you still hear me? Okay. All right, folks. So let's get started. And today we are actually covering two labs. So we have lab 10 and lab 11. So which means that we have two in-lab quizzes that we need to do today. And there are three different in-lab activities we're going to be doing. Okay. Now let me share my screen with you. All right, guys. So lab 10 is going to be about the blood. So we're going to discuss about blood. We're going to look at the components of blood. We're going to look at the different types of cells we have in the blood. And we're also going to discuss about blood group type, pulse, and blood pressure. Then lab 11, we're going to be discussing about the heart. Okay. Now, both of these topics, we have already discussed them in the lecture, so it's going to be a review for most of you that are taking my lectures as well. So let's start with the components of blood first, folks. What do we find in our blood? Look, out of our blood, most of our blood is actually made of this clear fluid called the plasma. Now, on average, a human has about 5 liters of blood. Males have a little bit more, and females have a little bit less, okay? But on average, total is about 5 liters. Now, 55% of our blood is the plasma, and within the plasma, most of the plasma is actually water. So it's 90% water and 10% solutes dissolved within the water. Now, the rest of the blood is what we call the formed, excuse me, the formed elements. And the formed elements are a fancy name for what we call the cells. So these are the blood cells. In between the plasma and the bottom of the tube here with the red stuff, we're going to have a cream colored layer called the buffy coat. And within the buffy coat, this is where we're going to find our white blood cells and our platelets. Then the rest of the blood is going to be made of cells called the red blood cells, also called the erythrocytes. Okay? As you can tell right here, look, folks, just by looking at the slide here, we have a lot more erythrocytes or red blood cells than we have white blood cells and platelets. The red blood cells are the most abundant blood cells in our body. Now let's take a closer look at the formed elements at the blood cells and see what they do. What is the main function of each one of them? The red blood cells' main function is to carry oxygen gas and distribute the gas throughout the entire body. Now the red blood cells are also called the erythrocytes. Erythro means red. Cyte means cell. So these are the red blood cells. The other group of cells that we have in our blood are the white blood cells, and the white blood cells are also called the leukocytes. Leuko means white, cyte means cell. Now, what is the main function of the leukocytes? The main function is for our immune protection. Within the white blood cells, which we have 5 different types, they are going to help protect us against anything that is foreign to our body. Bacteria, viruses, etc. Okay? And then the rest of the formed elements are made by what we call the platelets, and the platelets are also called the thrombocytes. Thrombo means clot, cyte means cell. So these are the cells in our body, in our blood, that helps with the process of blood clotting. Okay? Two very important terms to know and understand. One is a hematocrit, the other one is anemia. A hematocrit is actually a test that we do with our blood to check for the percentage of red blood cells in our blood. I guarantee you, if you ever had a blood test done, one of the tests that they're going to do with your blood is to do a hematocrit test. Females have a normal range between 38 and 46% of the blood will be red blood cells in terms of volume. And the males, in terms of the volume of the blood, 40 to 54% is going to be the erythrocytes, the red blood cells. Okay? Now, what if you do a hematocrit and your result is lower than the range, than the normal range? Now you are officially anemic, because anemia is actually a decreased capacity of our blood to carry oxygen gas. And anemia can be caused by several different things. It could be because of a low level of a red blood cell. It could be because you don't have enough hemoglobin within your red blood cell. It could be because you don't have enough vitamin B12 to allow for new red blood cell formation. So several different things can lead to anemia, okay? Now, the process of making new blood cells is a process that we call hematopoiesis. And hematopoiesis happens inside our red bone marrow. So inside the red bone marrow, we have little stem cells called the hemocytoblast. So what is a hemocytoblast? A hemocytoblast is a blood stem cell. And we have discussed about stem cells before. Stem cells, they are special because they can differentiate, they can transform in other cells in the body. So the hemocytoblast can differentiate into a lymphoid stem cell or a myeloid stem cell. Lymphoid stem cells are going to become our lymphocytes, the T cells and the B cells, while the myeloid stem cells, they're going to become the other blood cells. They're going to become the erythrocytes, the platelets, and some of the other white blood cells. Okay? All right, folks. Next, I want to talk about blood group type. And one of the activities we're going to be doing today is related with blood typing. And if you guys don't understand this, you're going to have a hard time doing the in-lab activity. Okay? So I need your full attention. And if something is not clear, guys, please stop and ask me. So when we talk about blood group type, there are two different blood group types that are relevant and important for humans, especially in the context of a blood transfusion. We have what we call the ABO blood group type, and we have what we call the RH blood group type. First of all, they are independent from one another. Okay? Second, when we talk about blood group type, we are specifically talking, about the presence or absence of specific surface markers on the surface of the red blood cell. Now, another name for surface markers are what we call the antigens. So antigens or surface markers, they mean the same thing. Okay? Now, in regards to the ABO blood group type, your blood group type is going to depend on the presence or absence of two different antigens. We have the A antigen and we have the B antigen. So let's see here how the presence or absence of those antigens make our ABO blood group type. If you have the A antigen on the surface of your red blood cell, you are going to be type A. If you have the B antigen on the surface of your red blood cell, you're going to be a type B. If you have both the A antigen and the B antigen on the surface of your red blood cell, you're going to be a type AB. And if you don't have antigens against the A or the B antigen, If you don't have antigens for the A or the B antigen, then you're going to be what we call a type O. Okay? That's it, guys. That is as simple as it gets. If you have an antigen, you are that type. If you don't have the antigens, you're going to be a type O. Now, what about the Rh blood group? The Rh blood group is even simpler. If we have the Rh antigen on the surface of the red blood cell, you're going to be Rh positive. If you don't have it, you're going to be Rh negative. So you can be a type A positive or negative, type B positive or negative, AB positive or negative, and also a type O positive or negative, because again, they are independent. Okay? Now, when we talk about the blood group type and blood transfusion, and this is what gets a lot of students, and they have a hard time understanding, is that not only we need to worry about the antigen on the surface of the red blood cell, but we also need to be mindful of the antibody in the plasma of that individual. So what is an antibody? What is the difference between an antibody and an antigen? Guys, antigens are proteins on the surface of the red blood cell. Okay, remember, they are surface markers. They are stuck to the surface of the cell. Antibodies, they are proteins circulating freely in our blood. They are not attached to anything. First of all, that is one difference. Another difference is that antibodies bind to antigens and they lead to the destruction of the cell. When an antibody finds its own antigen and binds to it, it will lead to the destruction of that cell. Antibodies are a protective mechanism that we have in our body. Because if something comes into our body that does not belong to our body, the antibody, specific antibody, is going to bind to antigens on the surface of whatever comes into our body and is going to lead to its destruction. Okay? So, if you are a type A, an ABO blood group type A, and you have the A antigen on the surface of the red blood cell, automatically, you are born that way, you will have antibodies against the B antigen, what we call anti-B antibody. If you are a type B and you have the B antigen on the surface of your red blood cell, you're going to have antibodies against the A antigen. So you're going to have anti-A antibody. If you are a type AB, you will not have antibodies against the A or the B antigen, so none. And if you are a type O, meaning you have no antigens on the surface of the red blood cell, you will have in your plasma antibodies against both the A antigen and the B antigen. Okay? What does that mean? It means that if you are a type O and a type B donates red blood cells to you, the antibodies will bind to the antigen and will lead to the destruction of that red blood cell. This is what we call agglutination and hemolysis. So agglutination is the clumping of the red blood cell. Hemolysis is the breakdown of the red blood cell. Okay? Now, with that in mind, let's bring the concept of universal donor of blood and universal recipient of blood. So who can donate blood to anybody? Who can receive blood from anybody? All right, and we're going to specifically talk about the ABO blood group type now. So who is a universal donor? A universal donor is a type O. Why? Guys, look at the red blood cell of a type O. A type O red blood cell has no antigens for A or B. So there is no A antigen, there is no B antigen. What does it mean? It means that this individual here can donate blood to anybody. It does not matter what antibody you have in your plasma. Because if there are no antigens for the antibody to bind, the red blood cells are going to be left alone. They're not going to be destroyed. Okay? Now, specifically, O negative is what we consider a universal donor. It can literally donate blood to anybody. What about universal recipient? Universal recipient is a type AB. This guy right here. Why? Because the type AB, if you look over here, guys, this individual has no antibodies in the plasma. There is no antibody against the A antigen or against the B antigen. So it doesn't matter who donates blood to this individual, because this individual has no antibodies to bind to the antigen. Therefore, the red blood cells are going to be left alone. Okay, they're not going to be lysed, they're not going to be agglutinated. Now, specifically, AB positive is what we consider a universal recipient of blood. You can literally receive blood from anybody. Okay? So, help me out here, folks. Let's do a blood typing. By the way, this is what you're going to be doing in one of the activities we are doing today. You are going to be typing blood of different patients, okay? Different individuals. So here we have blood from 4 different individuals. We don't know the blood type of those individuals. We're going to find out right now. We're going to test the blood type right now. What do we need? All that we need are samples of untreated blood. We need a serum that contains the anti-A antibody, meaning antibody that can react to the A antigen. We need a serum that contains the anti-B antibody, meaning antibody that reacts to the B antigen. And we also need a serum that contains the anti-RH antibody, okay? Which is the antibody that reacts with the RH antigen. All right, so let's try the anti-A antibody first. So I'm going to add a drop of the serum to each one of the untreated blood samples, right there. So we can see here that individual number 1 and individual number 3, something happened with the red blood cells. All the blood cells got destroyed and now they're all clumped together, guys. But we can see here nothing happened for individuals number 2 and number 4. All right, so now I'm going to test the anti-B serum. So I have a fresh sample of blood. I'm going to add a drop of the anti-B serum, stir it, and see what happens. There we go. Nothing happened for individual number 1 and 4, but we have agglutination and lysis for individuals number 2 and 3. So now I can already tell what is the ABO blood group type of those 4 individuals. Individual number 1 is a blood type A. Why? Well, that is because when I added the antibody against the A antigen, the antibody recognized the antigen, bound to the antigen, and destroyed the red blood cells. But when I added the anti-B antibody, nothing happened, which tells me that this red blood cell does not contain the B antigen, only the A. Okay? What about individual number 2? And the individual number 2 then, by the same token, is going to be a type B, because only the anti-B serum reacted. Individual number 3 is going to be a type AB, because both serums reacted with the red blood cells. And individual number 4 is going to be a type O, because none of the anti-serum and the antibodies reacted, which means there are no antigens in the surface of those red blood cells. Okay, now what if I want to test for the RH? Well, all that I have to do again is get a fresh sample of blood, and I'm going to add a drop of the anti-RH serum. If, when I add the serum, it reacts with the red blood cell, this is going to be RH positive. If it doesn't react, it's going to be RH negative. Okay? Any questions, guys? All right, now let's take a closer look at the RH antigen. And RH antigen is especially relevant, folks, when we have a mother who is RH negative, meaning that her red blood cells have no RH antigen, and she's carrying a baby, and the baby is RH positive. So the mother is negative, the baby is positive. Now, what's going to happen here is that the blood of the mom and the baby will mix when the baby is in the uterus, is growing. And the mom now will start developing antibodies against the RH antigen. So she will develop anti-RH antibody. Now, in the first pregnancy, nothing's going to happen. This baby is going to be fine because it takes enough time for enough antibody to be produced. So by the time that she has enough anti-RH antibodies, the baby is going to be born. It's going to be out of the womb. Okay? But let's say the same mom gets pregnant the second time, and again, the baby is RH positive. Now, this is a serious issue. Not for the mom herself, but actually for the fetus. Why is that serious? That is problematic because now the mom has antibodies against the RH antigen. And the antibody is going to find the RH antigen, is going to bind to the red blood cells, and is going to lyse and destroy the red blood cells of the fetus. This is going to lead to a condition called the hemolytic disease of the newborn. Okay, this is very serious, and it could kill that fetus. It could kill that baby. Now, this is not such a big issue anymore because we have treatment for that. When the doctor finds out that the mom is RH negative and the baby is RH positive, after the first pregnancy, after the baby is born, the doctor will give shots of this serum called RhoGAM to the mom. So the mom is going to receive some shots of this immune serum called RhoGAM. What does the RhoGAM does? The RhoGAM is going to capture the RH antigen circulating in the mom's blood and is going to prevent the formation of the antibodies. So the mom will have no antibodies against the RH antigen. So if she gets pregnant the second time, it's not going to be an issue at all for the baby because she doesn't have any antibodies against the antigen. Okay. So guys, let's watch this video here, which is going to summarize and talk a little bit more about the ABO and the RH blood group type.
Covering your guacamole with this citrus juice and sealing it with plastic wrap and putting it in the fridge can help preserve it for longer. Is it lime juice?
A blood type or blood group is a means of classifying blood based on the presence of specific antibodies and on whether red blood cells have specific inherited antigenic substances on their surfaces. There are 38 of these blood group systems based on various antigens. These antigens can be proteins, carbohydrates, glycoproteins, or glycolipids. The most important blood group systems are those that denote ABO and RH status, since these determine suitability of blood for transfusion. We will be discussing them in this video. The ABO blood group system involves 2 antigens and 2 antibodies found in human blood, antigen A and antigen B, and antibody A and antibody B. The A and B antigens are found on red blood cells, while antibodies A and B are in the serum. The antibodies are usually IgM, or immunoglobulin M, antibodies. Humans have one of 4 combinations of antigens, along with a corresponding combination of antibodies, which we'll get to next. People with blood type A have antigen A, people with blood type B have antigen B, People with blood type AB have both antigen A and antigen B, and people with blood type O have neither antigen. If you're blood type A, then you have antibody B. If you are blood type B, you have antibody A. If you are blood type AB, you have neither antibody. Finally, if you are blood type O, you have both antibody A and antibody B. If antibody A encounters antigen A, or if antibody B encounters antigen B, an agglutination reaction results. This is very dangerous, and we'll cover this in a follow-up video. Blood transfusion is safe only as long as the recipient's serum doesn't have antibodies for the donor's blood cell antigens. The second most significant blood group system for blood transfusion is the Rh system. Rh stands for rhesus, and it involves 50 antigens. The most significant Rh antigen is the D antigen, as it's the most likely to cause the immune system to react. The presence of D antigen is signified by a plus sign after the ABO blood type. Lack of the D antigen is signified by a minus sign. For example, someone who has B antigen and Rh antigen would be signified as B positive. It's common for someone who is D negative to not have anti-D antibodies, but these people can develop these antibodies after a sensitization event. This can happen, for instance, during a blood transfusion, or during a fetal-maternal transfusion of blood during pregnancy. This can result in the development of Rh disease. So a D-negative patient without sensitization to D-positive red blood cells can receive a transfusion of D-positive blood once. Then, they become sensitive to D antigen, so a future transfusion of D-positive blood would result in a dangerous transfusion reaction. And, in the case of D-negative females without prior sensitization, receiving D-positive blood results in a risk of hemolytic disease of the newborn. Hence, Rh D-positive blood is not given to D-negative women of childbearing age. Meanwhile, D-positive patients do not react to D-negative blood. The same kind of matching is done for other Rh antigens. Now let's apply what we've learned to fill out this red blood cell compatibility table. O negative is the universal donor. Since there are no A, B, or D antigens, O negative can donate blood to anyone without triggering an immune response from A, B, or anti-D antibodies. AB positive is the universal receiver, since there are no antibodies that will trigger an immune response to A, B, or D antigens. Blood plasma is the liquid component of blood which holds blood cells and other constituents of whole blood in suspension. This is where you find A, B, or anti-D antibodies. Hence, plasma compatibility is the inverse of red blood cell compatibility. Someone with blood type O can receive plasma from any blood type, while someone with blood type AB can only receive plasma from other people with blood type AB. Someone with blood type O can only donate plasma to others with blood type O, while those with AB blood can donate plasma to anyone. It must be noted that with rare exceptions, an individual has the same blood group for life. On rare occasions, it can change through addition or suppression of an antigen during infection, malignancy, autoimmune disease, or due to a bone marrow transplant. If someone with a different ABO type donates bone marrow to you, your blood type will eventually convert to the donor's type.
All right, folks, any questions, guys? All right, very quickly here, let's talk about pulse and blood pressure. We have different points within our body in which we can check our pulse, and our pulse is our heart rate, right? Every time that our ventricles contract, especially the left ventricle, and it's going to push blood out of the left ventricle, now the blood is going to go into the arteries, which is going to increase the pressure in the arteries, and we can feel the pulsation in different superficial arteries. Now, one of the arteries that we use to check our blood pressure is what we call the brachial artery. And we use the brachial artery with a device that is called the sphygmomanometer. Now, what is a blood pressure, guys? First of all, blood pressure is the pressure that blood applies to the inner walls of the artery. So I'm going to repeat that. The blood pressure is the pressure that blood applies to the inner walls of the arteries. And when our heart contracts and the ventricle pump blood out of the heart, that blood will go into the arteries, which means now that we have more blood inside of the arteries. And the more blood you have in the arteries, the higher the pressure. So this is what we call the systolic blood pressure. And a normal systolic blood pressure is 120 millimeters of mercury. Now, when the heart relaxes and now the ventricle refill with blood, there will be less blood in the arteries because the blood is going inside of the heart. So if you have less blood in the artery, you're going to have a lower pressure. And this is what we call the diastolic blood pressure. And a normal diastolic blood pressure is 80 millimeters of mercury. Okay, so a normal blood pressure is 120 over 80. Again, 120, the number on top is the systolic blood pressure. The number at the bottom is what we call the diastolic blood pressure. So how do we use this sphygmomanometer to check our blood pressure? What we're going to do is that the sphygmomanometer includes a cuff with a balloon and a little bulb in which we can pump air inside of the balloon. And there is a gauge attached to the balloon, which is going to tell us the pressure inside of the balloon. And again, the unit of pressure is millimeters of mercury. Now, we're going to take this cuff and we're going to put it into the upper arm, wrapped around the upper arm, and we are going to raise the pressure in the cuff by pumping air into the cuff. And we're going to raise the pressure usually around 160 millimeters of mercury, okay? Because we want to make sure that we are going to completely stop the flow of blood through the brachial artery. So now what we're going to do is that we're going to take a stethoscope and we're going to put the diaphragm of the stethoscope right over the brachial artery. And we are going to slowly let the air out of the cuff. Now, as the air is out of the cuff, we can see here in this graph that the pressure inside of the cuff starts to go down. When the pressure inside of the cuff is the same as our systolic blood pressure, guys, we're going to hear the first of what we call the Korotkov sound. What is this Korotkov sound? The Korotkov sound is the sound of blood tumbling, struggling to pass through the brachial artery because there's still pressure on the brachial artery. So we're going to keep hearing the Korotkov sound as we let the air out of the cuff. We can see again that the pressure inside of the cuff keeps going down until when the pressure inside of the cuff is the same as the diastolic blood pressure. And now we're going to look at the gauge right here and we're going to see where the needle is. And this is going to be our diastolic blood pressure. And at this point right here, we're going to hear the last Korotkov sound. From then on, we don't hear anything anymore. Okay? And that is going to be the indication of our diastolic blood pressure. So once again, a systolic blood pressure is the peak of ventricular contraction. Diastolic is the pressure generated when the ventricles are relaxed. Okay. All right, guys, so we're done with lab 10. Let's go straight into lab 11, which we're going to be discussing about the heart. So we're going to take a look at the anatomy of the heart. We're going to take a look at the coronary circulation, heart sound, ECG, and then we're going to go over the lab activities. Folks, there are several things I'm going to be discussing right now in regards to the heart anatomy that will be on our next lab exam. Okay, so I need your full attention, and I hope that all of you guys already start to study for the next lab exam. So let's start with the basics here. Where is our heart located? Our heart is located within the thoracic cavity. About 2/3 of the heart is within the left side of the thoracic cavity, but about 1/3 of the heart is actually on the right side. So our heart is not all on the left side. Okay? Most of it is on the left side, but a little bit of it is on the right side of the thoracic cavity. The heart is positioned in a location within the thoracic cavity called the mediastinum, And our heart has a triangular shape to it. Okay, so we can draw a line here and we can see that it looks like a triangle. Now, just like any triangle, the apex of the heart right here is going to be pointing towards the left side of the body, and the base of the heart is going to be pointing towards the right side of the body. And this is very important because several of the structures of the heart, they are bilateral. And you need to know how to split the heart into two halves. How do we do that, guys? To split the heart into two halves, we're going to pass an imaginary line that is going to go from the base, is going to pass through the walls in between the ventricles, what we call the interventricular walls, and then is going to exit through the apex, just like this. So this side right here is the right side of the heart. This side right here is the left side of the heart. Okay? So let's take a closer look at the anatomy of the heart. And our heart has 4 different chambers. But before we name them, once again, let's split the heart in half. Let's pass that imaginary line from the base, going through the interventricular wall, exiting through the apex. So we have the right side of the heart, we have the left side of the heart. So now we can name the 4 different chambers. So on the right side, we have the right atrium. On the left side, we have the left atrium. On the right side, we have the right ventricle. On the left side, we're going to find the left ventricle. Okay, so these are the 4 chambers of the heart. Now, each one of these chambers, guys, they have a specific function. And to understand the function of the different chambers, let's look at the blood vessels that are bringing blood in and out of the heart. And those major blood vessels are what we call the great vessels of the heart. Okay, they are big blood vessels that bring blood in and take blood out of the heart. Let's start with the right atrium right here. If you look at the right atrium, we can see there are 2 blood vessels supplying deoxygenated blood to the right atrium. We have a superior vena cava and we have an inferior vena cava. The superior vena cava is going to bring deoxygenated blood from the neck and the head and then send the blood into the right atrium. The inferior vena cava is going to bring deoxygenated blood from the rest of the body and dump all of the blood into the right atrium. So what is the function of the right atrium? The right atrium receives all the deoxygenated blood coming from the body cells and tissues. Okay? From the right atrium, the blood is going to move into the right ventricle. And then from the right ventricle, now the blood is going to move into what we call the pulmonary trunk. Now, where is the pulmonary trunk? The pulmonary trunk is this section right here, guys. So this section of this blood vessel, this is called the pulmonary trunk. And this pulmonary trunk is going to split into 2 branches. One going towards the right, the other one going towards the left. These branches right here are what we call the pulmonary arteries. On the left side, we have the left pulmonary artery. On the right side, we have the right pulmonary artery. What are the pulmonary arteries going to do? The pulmonary artery is going to take the deoxygenated blood and is going to send the blood all the way to the lungs. The left pulmonary is going to send it to the left lung. The right pulmonary artery is going to send to the right lung. Now, once the blood is going to become again oxygenated in the lungs, the blood needs to return to the heart. And it's going to return to the heart through what we call the pulmonary veins. And we have, again, the right pulmonary veins, and we have the left pulmonary veins. Okay, remember the difference between an artery and a vein. Arteries take blood away from the heart. Veins bring blood back into the heart. So now all the oxygenated blood that is coming from the lungs is going to arrive in the left atrium. From the left atrium is going to go into the left ventricle, and now is going to enter different sections of the aorta. The first section of the aorta is what we call the ascending aorta, in which the blood is moving upwards. Then we have the aortic arc, also called the arc of the aorta, in which the blood now is going to make a turn. And then from the arc of the aorta, now the blood is going to enter the descending aorta, in which that oxygenated blood is going to go all over the body. Warning, guys. In a quiz like the one you're going to take today and in the exam, if I am pointing to a section of the aorta, you need to be specific. It is not enough just to say aorta. You need to tell me if I'm pointing to the ascending aorta, to the aortic arc, or to the descending aorta. Okay? Now, the heart is made of three different layers. The heart wall is made of three different layers. We have an inner layer called the endocardium, a middle layer called the myocardium, and an outer layer called the epicardium. The endocardium is covering the walls of the chamber. Excuse me. You know, the different chambers, the atria and the ventricles, the walls of the chamber are covered with that endocardium layer. The middle layer is the thick layer made of cardiac muscle cells. This is why we call it myocardium. Myo means muscle. And then the outer layer is the epicardium, which is the same as what we call the visceral pericardium. Remember that our heart is wrapped around a double-layered membrane, as we can see in the picture right here. If you look at this heart, guys, there is two layers surrounding the heart. One layer that is touching the organ itself, the viscera, that we call the visceral layer, also called the epicardium. And then the other layer that is on top called the parietal layer, okay? In the middle, we have a cavity called the pericardial cavity, which is filled with pericardial fluid. Now, we have a different circulation of blood in our body. There are several different types. We have systemic circulation, pulmonary circulation, coronary circulation, cerebral circulation, fetal circulation, hepatic circulation. The main circulation of blood is what we call systemic. What does systemic mean? Systemic means all over the body. So the systemic circulation is the circulation of blood between the heart and the rest of the body. While a pulmonary circulation is the circulation of blood between the heart and the lungs. We can see that in the systemic circulation, the arteries are labeled in red because they are carrying oxygen-rich blood. And the veins are blue because they are carrying oxygen-poor blood. But in the pulmonary circulation, it is the opposite. In the pulmonary circulation, the arteries are blue and the veins are red. Okay, because now the arteries are carrying oxygen-poor blood, which is going to go to the lungs to become oxygenated, and the veins are carrying oxygen-rich blood, which is returning to the heart. Now, let's talk about the pulmonary circulation. What is the pulmonary circulation? The pulmonary circulation is the circulation of blood to the heart walls to feed the cardiac cells with oxygen and nutrients. And we have 6 coronary arteries that you guys need to know for the exam and 3 coronary veins. Guys, this will be in our next lab exam. Guarantee you. So you need to be able to identify and name the 6 coronary arteries, which we're going to go through right now, and the 3 coronary veins, which we're going to discuss in the next slides. Okay, so everybody's attention here, please. All right. First of all, let's split the heart into two halves once again. So we're going to pass that imaginary line from the base, going through the interventricular wall, exiting through the apex. So here we have the right side and the left side of the heart. Now, if you look on the right side of the heart, we have a blood vessel right here that is literally siphoning blood from the ascending aorta on the left side. On the right side. This is what we call the right coronary. If you look on the left side, we're going to see another blood vessel siphoning blood from the ascending aorta on the left side. This is what we call the left coronary. Now let's follow the right coronary and the left coronary, okay? The right coronary, when it gets to this point right here, will split into two major branches. One going towards the front of the heart. This is what we call the marginal branch. The other one going towards the back of the heart. This is what we call the posterior interventricular branch. Let's follow the left coronary now. When the left coronary gets to this point right here, it's going to split into two major branches. One in the front called the anterior interventricular branch, and the one going towards the back side of the heart. This is what we call the circumflex branch. Okay? So, do not forget to take a look at the coronary arteries. Let's recap here. The right coronary splits into marginal branch in the front and a posterior interventricular branch in the back. The left coronary splits into the anterior interventricular branch in the front and the circumflex branch in the back. Okay? Now let's take a look at the coronary veins. And as I said, there are three coronary veins I want you guys to know by name for the exam. The major one right here in the front of the heart, guys, running next to the anterior interventricular branch, this is what we call the great cardiac vein. So it's in the front of the heart. This great cardiac vein now is going to go towards the back of the heart, and is going to become what we call the coronary sinus. And then this coronary sinus is going to branch off towards the apex of the heart, in the back of the heart, and it becomes what we call the middle cardiac vein. Okay, so these are the three coronary veins. The great cardiac vein in the front, coronary sinus going towards the back, and then a middle cardiac vein going towards the apex of the heart, in the back of the heart. Okay? So again, let's just, let me show you all of these blood vessels of the coronary circulation, both in the anterior veil of the heart as well as a posterior veil. So we can see the right coronary splitting into marginal artery and posterior interventricular artery. The left coronary splitting into the anterior interventricular artery and the circumflex artery going towards the back. And then we can take a look at the veins. Here in the front, we have the great cardiac vein. And then we have the middle cardiac vein, which is in the back of the heart. And also going towards the back of the heart, we have the coronary sinus. Okay? All right, guys, so we talked about the heart chambers, we talked about the great vessels of the heart, we talked about the coronary circulation. Now let's take a look at the heart valves. Just like the chambers, we also have four different heart valves. We have two called atrioventricular valves, and we have two semilunar valves. Before we name the atrioventricular valves or the AV valves, let's split the heart into two halves. Right there. So now I can name them. On the right side, I have the right AV, which is right between the right atrium and the right ventricle. On the left side, I have the left AV, which is in between the left atrium and the left ventricle. Now, the right AV valve, folks, is also called the tricuspid valve, while the left AV has two other names. It's also called bicuspid or called mitral. Everybody pay attention here, please. For the exam, pick one name and one name only. It is not the right AV tricuspid valve. It is not the left AV bicuspid mitral valve. Adding more names doesn't give you extra points. Pick one name. Okay? The other thing that I want to mention that is very important, you either say right AV or you say tricuspid. There is no right tricuspid. If you put down right tricuspid, I'm going to take points away from you. Because if you say right tricuspid, you imply there's a left tricuspid. There is no left tricuspid. Same thing for the left AV. You either say left AV, or you say bicuspid, or you say mitral. There is no left bicuspid. There is no left mitral. All right? And now the AV valves, they have these little strings right here, which helps with the opening and closing of the AV valves. These strings are what we call the chordae tendineae. And this chordae tendineae, they are bilateral. If I am pointing to these ones right here, these are the left chordae tendineae. If I'm pointing to these ones right here, these are the right chordae tendineae. And the chordae tendineae, they anchor themselves to tiny little muscle. They attach themselves to tiny little muscles in the chambers, in the ventricles. These little muscles are called the papillary muscles. And once again, they are bilateral. If I'm pointing to these guys here on the exam, these are the left papillary muscles. If I'm pointing to these ones right here, they are the right papillary muscles. Okay? Now, what about the SL valves, the semilunar valves? We have two different types. We do not have right and left SL valve. We have the pulmonary SL and we have the aortic SL. The pulmonary SL is going to separate the right ventricle from the pulmonary trunk. This is why it's called pulmonary SL, and it's shown here in blue. The aortic SL, shown in red right here, is going to separate the left ventricle from the ascending aorta. This is why it's called aortic SL valve. Okay? The SL valves do not have a chordae tendineae. Okay? All right, folks, we are done with the anatomy of the heart. So everything that you need to know for the exams in terms of anatomy, we have already discussed. Okay? Let's move on now and talk a little bit about the physiology of the heart. How does our heart beat? And the reason why our heart can beat on its own is because our hearts, they have a cluster of very special cells called the natural pacemaker cells. What is so special about these cells? These cells, they can generate their own action potential. What is action potential, guys? Action potential is electrical impulse. Okay? And when the action potential is generated, that is going to lead to the contraction of the cardiac cell. And we're going to talk more about that in a minute. Now, the natural pacemaker cells of the heart, they are going to cluster into two little nodes inside the right atrium. There is the main node called the sinoatrial node, or the SA node, and then there's another node right by the right AV valve, and this is what we call the AV node, or the atrioventricular node. Can you guys see that the arrows spreading from the SA node? These arrows right here, guys, they're representing action potential. Okay? So the AV node, the SA node, they produce their own electrical impulse, and then they're going to send those electricity, the electrical impulses, through the rest of the heart wall. And this is how our heart is going to contract in a very coordinated manner. We're going to start with the contraction of the atria first, both of them at the same time. Then we're going to have the contraction of the ventricles, both of them right and left at the same time. Now, the AV node right here is going to take the signal, the action potential coming from the SA node, and is going to send the signal inside the ventricular walls. We can see there is several segments of nerves right here, which will spread the action potential through the ventricular wall. The first section of the nerve is what we call the bundle of His. Then the bundle of His is going to split into two, what we call the bundle branches. So we have a left bundle branch and we have right bundle branch. And the bundle branches get all the way to the apex. Now they're going to become what we call the Purkinje fibers. Okay? And again, they are spreading the action potential through the cardiac walls. A little bit about the heart sound. Our heart's sound makes a very peculiar noise. It's what we call the lub-dub sound. And it goes like this. And the lub sound, also called the S1 sound, is due to the closing of the AV valves, both of them, right and left at the same time. So after the blood passes from the atria into the ventricles, through the AV valves, now the AV valves are going to shut close, preventing the backflow of blood from the ventricles into the atrium. And the closing of the AV valves cause the lub sound. Now, what about the dub sound? The dub sound, also called the S2, is due to the closing of the SL valves, both of them, pulmonary and aortic, at the same time. Okay? And finally, let's take a look at the ECG. What is ECG, guys? ECG stands for electrocardiogram. And an ECG is going to give us information about the health of our heart, how well our heart is beating. And it's going to tell us about the cardiac cycle. Okay? Now, what we're looking at right here on the screen, this is the result of an ECG showing a single heartbeat, which is about 0.8 seconds. That's how fast a single heartbeat is. And we can see here there are these different waves within the ECG. What is the first wave right here? This first wave, called the P wave, this represents atrial depolarization. What is atrial depolarization? First of all, depolarization means the opening of the sodium channels in the cells of the atria, allowing sodium to enter the cell and leading to the formation of a new action potential. That's what depolarization means. So what we see right here is that the cells of the atria, they start to depolarize and they start to spread that action potential from the SA node towards the AV node. Okay? Immediately after atrial depolarization, we're going to have an atrial contraction. The atria will contract and squeeze blood, push blood into the ventricles. Next, we have the R wave, and the R wave represents ventricular depolarization. So now the cells of the ventricles are going to depolarize. Okay? And then finally, we have the T wave, and the T wave represents ventricular repolarization. What is repolarization? Repolarization is when the sodium channels close, the potassium channels open in the cells, and now potassium is going to leave the cell and is going to allow the cell to go back into the resting membrane potential. So Immediately after ventricular repolarization, we are going to have ventricular relaxation. So it goes this way, guys. We have a depolarization followed by contraction, repolarization immediately followed by relaxation. Okay? So let's watch this video right here, which is going to summarize the ECG and the cardiac cycle.
It's Google Gemini with the assist.
Foodland $6 Eats, your favorite local grinds for just $6 every Friday, only at Foodland.
The cardiac conduction system consists of the following components. The sinoatrial node, or SA node, located in the right atrium near the entrance of the superior vena cava. This is the natural pacemaker of the heart. It initiates all heartbeat and determines heart rate. Electrical impulses from the SA node spread throughout both atria and stimulate them to contract. The atrioventricular node, or AV node, located on the other side of the right atrium near the AV valve. The AV node serves as electrical gateway to the ventricles. It delays the passage of electrical impulses to the ventricles. This delay is to ensure that the atria have ejected all the blood into the ventricles before the ventricles contract. The AV node receives signals from the SA node and passes them onto the atrioventricular bundle, AV bundle, or bundle of His. This bundle is then divided into right and left bundle branches, which conduct the impulses toward the apex of the heart. The signals are then passed onto Purkinje fibers, turning upward and spreading throughout the ventricular myocardium. Electrical activities of the heart can be recorded in the form of electrocardiogram, ECG, or EKG. An ECG is a composite recording of all the action potentials produced by the nodes and the cells of the myocardium. Each wave or segment of the ECG corresponds to a certain event of the cardiac electrical cycle. When the atria are full of blood, the SA node fires, electrical signals spread throughout the atria and cause them to depolarize. This is represented by the P wave on the ECG. Atrial contraction, or atrial systole, starts about 100 milliseconds after the P wave begins. The PQ segment represents the time the signals travel from the SA node to the AV node. The QRS complex marks the firing of the AV node and represents ventricular depolarization. Q wave corresponds to depolarization of the interventricular septum. R wave is produced by depolarization of the main mass of the ventricles. S wave represents the last phase of ventricular depolarization at the base of the heart. Atrial repolarization also occurs during this time, but the signal is obscured by the large QRS complex. The ST segment reflects the plateau in the myocardial action potential. This is when the ventricles contract and pump blood. The T wave represents ventricular repolarization immediately before ventricular relaxation, or ventricular diastole. The cycle repeats itself with every heartbeat.
All right, guys, any questions? All right. So, what are we doing today? So today we have three different activities, okay? Two of the activities are related with Lab 10, and one activity is related with Lab 11. So we have the virtual blood typing and the blood pressure on Edpuzzle. This is a video, which is going to be related to Lab 10. And then the heart model video and Edpuzzle is going to be for Lab 11. Now, you're all familiar with the Edpuzzle. I don't need to go through that, but I do want to go through the virtual blood typing exercise and explain to you what you need to do. So let me share my screen with you here for a second. All right, guys, so if you go to our Lamaku site and you go over Lab 10 in-lab activities and you click on that, so you're going to have two parts, part 1 and part 2. If you click on part 1, that is going to take you to the Edpuzzle, which you're going to watch the video on blood pressure, how to measure blood pressure using a sphygmomanometer. When you click on the lab activity part 2, you're going to get to this Word documents, right? That should be here. Hold on a second. You can also click on the download, and then the document is going to open. Okay? So this is what the document looks like. So this is a virtual blood typing transfusion lab simulation lab. What do you have right here, folks? This is where you're going to be filling up all the information, all the questions that are here in this pre-lab part, and then in the lab simulation section. Where is the lab simulation? The lab simulation, what you're going to do is that you are going to click on this link right here. Let me just copy and paste here. And when you click on that link, this page is going to open up. And this is your blood typing activity. All right? It's a very simple activity. What are you going to be doing? You are going to be typing the blood of different individuals. So you're going to be reading here about each one of the individuals. Then you're going to click next. Then you're going to continue. You're going to follow the instructions and continue the activity. After you're done with the activity and you fill in the documents for the blood typing documents, you're going to save that. You need to save that either as a PDF file or a Word file. If you have a Mac and you save it in any other way and you send it to me, I am not going to be able to open it. So save it as a PDF file or a Word document. Okay, and then you're going to email that the lab to me. You need to send it to me by the end of class today. Okay, so after you're done with the virtual blood typing and you watch the two videos on Edpuzzle, the one on blood pressure, the other one on the heart model, then you're going to jump on Lamaku and you're going to take the in-lab quizzes. And there are two quizzes open today, which is a quiz for Lab 10 and a quiz for Lab 11. And after you're done with the quiz, you're good to go. Does anybody have any questions on what they need to do today? All right, guys, let's get started.